physiochemical and biological properties of peptide drugs remain key challenges to development these days. The formulation of microspheres and nanoparticles for peptide delivery to enhance bioavailability and therapeutic efficacy is emerging as one innovative approach to address these challenges. Here at Bachem, we can support your successful microsphere and nanoparticle formulation by providing superior quality APIs, as well as with our in-house capabilities for specific studies on topics relevant to microspheres and nanoparticle formulations, such as, for example, surface area and aggregation studies. The Bachem approach to innovation also involves collaboration. Together with the German startup Biologics, we developed the Peptide Easy Clean platform, in short the PEC platform. This platform can enable the efficient purification and modification of complex peptides. So what is the PEC platform? PEC is a catch and release system used as an anchor for peptides, which enables chemical modifications, improves physical properties, and also allows for parallel purifications. PEC consists in a reductively cleavable linker tagged to a peptide which is introduced at the end of an SPPS. Amongst the advantages of the PEC technology, there are many. Firstly, PEC is universally applicable. It can also help increase the speed of peptide development because it allows for parallel purification in contrast to RP-HPLC, which is a linear approach. Further, PEC can facilitate the purification of difficult peptides. Thanks to the linker tag peptide, no aggregation, no stickiness, and increased solubility in organic solvents is achieved. In addition, PEC improves drug stability and efficacy through its functionality as a modification platform. The innovative safety release enables conjugation chemistry to explore the impossible through highly efficient solid-phase peptide modifications.
In the area of innovation, Bachem have signed a collaboration agreement with Jitsubo to get access to their molecular hydrogen technology. This technology resembles solid-phase peptide synthesis, but rather than have a resin to build up the peptide, it uses an anchor or tag, which is soluble in organic solvents and insoluble in aqueous solvents. This has the advantage of being able to avoid the use of certain hazardous solvents such as DMF, so therefore it opens the door for greener chemistry. It produces mainly aqueous waste. It uses a lower equivalent of FMOC amino acids, and all steps up to the precipitation of the product can be carried out in the same reactor. Bachem recently hosted a webinar where they introduced Enzymatic and their CEPS technology. This involves the ligation of peptide fragments using enzymes. Omnoligase is one such enzyme, which is highly selective. Enzymatic also have access to enzyme engineering technology where they can produce selective peptidoligases. This technology is especially used where stepwise peptide synthesis cannot be used because the peptide in question is either very long or difficult to manufacture. Examples of this are linear peptides, which are greater than 60 amino acids long, or certain cyclic peptides greater than 25 amino acids long. And these have been shown to be made efficiently using the CEPS technology from Enzymatic. We very much look forward to answering any questions that you might have on these technologies at Biologics Europe.
Given the decades of experience in the development, production, and regulatory support of APIs, Bachem is expanding its manufacturing platform from peptides to oligonucleotides. We expect the demand of oligonucleotides manufacturing to be fastly growing in the future. This is reflected in the strong interest in the therapeutic application of antisense technology and non-coding RNA biology for a broad range of indications. Bachem's aim is to become the first choice manufacturer of oligonucleotides. We offer process development and GMP manufacturing of oligonucleotides in a multi-hundreds of gram scale. Large-scale manufacturing will be made available later this year. We also offer innovation in equipment train, featuring engineering solutions for improved process control, dedicated R&D team with expertise in oligonucleotides chemistry, analytical chemists with long-standing experience in developing and validating test methods, and regulatory support by our in-house array specialists. Nowadays, it is more and more crucial to meet increasing demand while reducing our impact on the environment. Therefore, innovation in manufacturing and purification processes is our focus at Bachem. Our goal is to provide more productive, sustainable, and economic purification processes. Together with the Company for Life Science Equipment, Bachem implements a new chromatography method which allows us to provide high-quality peptide and oligonucleotide APIs in a much greener process. With this new purification method, we offer increased quality, higher yield for large-scale and GMP manufacture, increased efficiency, easier purification of complex molecules, and reduced solvent consumption. Very soon, we will be able to disclose further information about the new chromatography method at Bachem.
I think it's very important for our customers and future customers to understand that the decision by Bachem leadership to invest into oligonucleotide manufacturing capabilities was a very well prepared one. We have been lucky to forge strong partnerships on the customer side as well as on the supplier side, and have been able to identify the talent we need to accelerate our organizational learning. Despite having set ambitious goals, we still try to go step by step. So we're shifting our focus now from a first phase where we're focusing on closing the technology gap into a second phase where we build a solid foundation for future growth. Later, on our way to become a first-choice manufacturer for oligonucleotides, we hope to make our own contribution to advancing the field by making oligonucleotide API production more scalable and more cost-effective. Similar to peptides, the manufacture of oligonucleotides requires expert knowledge in solid-phase synthesis and protecting group chemistry. Downstream processing typically includes purification by chromatography and isolation by techniques like ultrafiltration, diafiltration, sometimes precipitation, and in almost all the cases, the final step is done by lyophilization. The manufacture of peptide APIs follows the exact same principle. And this is our core competence and our core platform that we have built over decades. Still, it's very important to understand, and we're very conscious about this, that there are important differences between peptides and oligonucleotides. The synthesis in flow-through columns consumes large volumes of solvents and reagents, for which our facility infrastructure will have to be appropriately expanded. Furthermore, oligonucleotides are negatively charged and as such highly water-soluble, requiring handling of aqueous solutions throughout the entire downstream process. Another very important difference to peptides. And let's not forget that oligonucleotide APIs, especially double-stranded entities, are considerably larger than peptides and pose challenges also from an analytical point of view. Overall, it is fair to say that our peptide manufacturing background and our analytical capabilities are certainly very helpful in our quest to build a successful oligonucleotide business.